Good morning. My name is Hayobami Adibite. I'm a doctoral candidate in the Bioenvironmental Science program at Morgan State University. Dr. Puntiwit McCarthy is my advisor, and today I'll be talking about the control of Neisseria meningitis, cerebral W capsule polymerase activity using the modified substrate. Neisseria meningitis is a gram-negative bacterium that is characterized by a bluish color when it is gram-stained. We have about 12 cell groups, and 6 pathogenic cell groups have been identified. These cell groups are characterized by the capsular polysaccharide, which is a non-virulence factor. They have a high immunogenic potential for vaccine development, and then these capsular polysaccharide protects the bacteria from external attack. It's also a known cause of virulence in kids and also immunocompromised adults. We have about 6 pathogenic cell groups that are in different parts of the world. In Africa, we have the prominent cell groups A, C, W, and X. And what are the approaches to containing Neisseria meningitis? Antibiotics have been used for the treatment of the infection, but vaccine is more effective because it prevents the disease from happening in the first place. The vaccines induce a protective immune response in the body against the pathogen represented in the vaccine. And then we have different vaccine types. We have the protein-based vaccine, the polysaccharide-based vaccine, and then the glycoconjugate-based vaccine. In this research, we are focusing on the glycoconjugate-based vaccine, and this is because they are more effective and they have a stronger lasting antibody response compared to other vaccine types. Talking about how this glycoconjugate vaccine works, we have the polysaccharide that is being hydrolyzed and chemically derivatized at the carboxyl or at the hydroxyl group before being attached to a carrier protein. This creates a random multi-point of attachment to the carrier protein, and research is moving towards the production of homogeneous-based vaccine. And because we want to understand the relationship between vaccine structure and immune response, it becomes very, very difficult because we have a random multi-point of attachment to a carrier protein. So this research is seeking to understand the relationship between vaccine structure and immune response by moving towards an homogeneous based vaccine because we want to know the particular site of attachment to the carrier protein. Talking about the vaccine gaps, the gap in knowledge, the study is focused on the heteropolymer because it contains repeats of two sugars, which is the cerebral W enzyme. Little is known about it. Not much has been done. We have many of the research being done in glycoconjugate vaccine development, especially for these Neisseria meningitis being done in cell groups A, C, Y, and also cell groups B. But we don't know much about the cerebral W enzyme. We need to be able to have a better understanding between vaccine structure and immune response, and that is why this research is very, very important. And talking about, what cell group W capsule polymerase looks like, we have the N-terminus that contains the galactosyl-transferase domain. We have the C-terminus, which contains the sialyl-transferase domain, and the intervening sequence, which little is known about. The capsule polymerase, which is found in this reaction scheme, is a free domain protein that catalyzes this reaction in the presence of an acceptor. The enzyme, which can be seen in this reaction scheme, takes galactose from UDP galactose and adds it to an acceptor. This same enzyme can then use the nucleotide donor sugar from CNP sire and then remove the salic acid and then add this to an acceptor. This is represented in this reaction scheme for us to have a better understanding of how this reaction works. The goals. Talking about the goals of this research is to be able to control sugars to move to a well-defined homogeneous glycoconjugate vaccine, and the goals have been we are using two methods in achieving these goals. One of the methods is the control of the synthesis of W-oligosaccharide using chain terminating nucleotide sugars. This will help us in understanding movement towards the homogeneous based vaccine. And then the second method involves the synthesize the conjugatable liposaccharide for conjugation using the copper free chemistry. And then what is click chemistry? Click chemistry, it contains a series of small molecules reaction that is commonly used in bioconjugation, and then it allows substrates of choice to be joined with specific biomolecule. For the two goals that I mentioned, we are using two methods. One of the methods involves the use of a fluorescent receptor, which is for MU galactose, that is enzymatically synthesized from for MU galactose and CMP salic acid in a reaction with W enzyme. And then this acceptor can be used to assess sugar composition and chain length of the product. This is the number one method used in this research to control the chain length. The other method involves, using the free click chemistry, which I said before, which means an azido modified disaccharide was used as an acceptor for the cell group W reaction, and then this azido group was used as a site of attachment to dibenzocyclotinamine dye via the copper free chemistry. The goal is for us to see if we can reduce the elongation using a potential competitive enzyme inhibitor, which is the CMP40 acetylcysteine, to control the chain length. These are the structure of the acceptor and then the dye as well. And then experimental procedures, just to quickly run through these, we perform growth, expression, and purification of the W enzyme and also CSF, which was used in this reaction. We have the SDS page gel for the CSS, which was used in the reaction to better understand the relationship, like I said before, between vaccine structure and immune response. And then this is the result. So the first result, which pertains to the first goal, we used the 4-MU galactose. We saw that the 4-MU galactose had a longer retention time, which means it has the potential to increase the chain elongation. Again, we're trying to move towards the production of homogeneous based vaccine. If we can increase or reduce the chain, we can better understand the relationship where the attachment is going on. Like I said before, we want to be able to understand the relationship, and then we want to be able to know the site of attachment to the carrier protein. Like conjugate vaccine, it is heterogeneous. Now, with the method being employed in this research, we are trying to know the precise place where the conjugation is going on, and then we want to be able to see how we can have a better understanding. And then the cell group W enzyme was used in setting up a copper free click chemistry reaction in the presence of DTT using the dibenzocyclotinamine. And then the cell group W enzyme, in the first method was used to extend the fluorescently labeled acceptor to assess the sugar composition and chain length of the product elongated. Like I said before, we see an elongation. So for the second method, for the second group, for the second goal itself, we see a decreased elongation of the acceptor in the presence of a potential enzyme inhibitor. From this figure right here, we see that the reaction has a shorter retention time. Again, we are using this process to make informed decision on how we can better assess the relationship between vaccine structure and immune response. So the two methodologies employed in this research and the acceptors used has clearly shown that we can potentially use 4-MU galactose and also CMP40 acetylcysteine as a potential competitive enzyme inhibitor to reduce the retention time and also to increase retention time, so that we can have a better understanding of the relationship between vaccine structure and immune response. So for future work, I want to continue to understand the potential activity of this enzyme inhibitor, which is the CMP40 acetylcysteine, using multi-12 mass spectrometry. Again, we're trying to understand the relationship, and being able to quantify this using mass spec will potentially help us to know if we actually made the simple for acetosire, which is the next phase in this experiment. And for this, I would like to say thank you to my advisors, Dr. Pumpty McCarthy, my committee members, and also Department of Chemistry in Morgan State University. Thank you so much.